So now, before we have the top 5 young investigators presentation, let us have a huge round of applause for all the young investigators who have submitted their abstracts. So can we have a huge round of applause for all of them? Thank you. So this was a very important part of our program. Out of 56 abstracts that we have received, the decision to selecting and finalizing top 20 was a difficult one. Such was the quality of the research work and content that we decided to invite the additional 15 more for displaying and showcasing their poster. After the morning poster evaluation session, the judges have shortlisted top 5 for oral presentation, and on the basis of their oral presentation, we will select our top 3. So for this, now let's have a huge round of applause for our top 5 investigators. Yes, can we have a huge round of applause for our top 5 investigators? So now for this, I would like to invite our chairperson, Professor Jyoti Tamang, Vice Chancellor officiating, and Senior Professor Microbiology, Sikkim Central University, Gangtok, Sikkim, and Professor J.B. Prajapati, Chairman ITA, Mumbai and Coordinator, SASNET Fermented Foods, Anand, Gujarat, India, for taking over the session. So let's have a huge round of applause for Professor Jyoti Tamang and Professor J.B. Prajapati. So each investigator has 7 minutes as a timer. Thank you.
Welcome to this very, very important session. We have 5 brilliant scholars who have come out after a very serious screening, and as informed, we'll have 7 minutes of presentation, and after that we can have 2-3 very, very relevant questions if there are. So I will first invite Mr. Rajdeep Dalal. He is from THSTI Faridabad, and his work is on gut commensal derived succinate impels colonic inflammation in ulcerative colitis. Rajdeep, 7 minutes. Okay.
Yeah, good afternoon, everyone. So first of all, I would like to thank to the organizing committee, Professor N.K. Ganguly and Nirja ma'am, for giving me this opportunity. And today I'm going to present my work under the title that is gut commensal derived succinate impels colonic inflammation in ulcerative colitis. So in this talk, I will cover the following question: how elevated succinates correlate the severity of the disease, how we can block this axis to ameliorate the IBD, and how we can design the novel consortia to engulf the accumulated succinates so that we can ameliorate the disease. So now, before going to the main course, I would just give the brief introduction. What is the IBD? So in IBD, actually, as per the clinician view, it's like there are lesions in the colon, and when we will extract these colons and we will do the analysis of the immune cells, there will be the cells there are of 2 types, the disease promoting cells and the disease preventing cells. So particularly in disease, when the dynamic will change to the pathogenic side, some more of disease promoting cells will be there. But if these cells are highly plastic, they can be converted back into the regulatory cells. But we need the modulators that can convert back into the regulatory cells. You know, the data is known that in the gonto mice, without microbiota, the maturation of Treg cells will never take place. So it means there is a potential to convert back into the regulatory cells, that is into the gut microbiota. So as per the microbiologist view, there are more than 100 trillion microbes are present. And these are the microbes, or they have as per the nutraceutical experts view, what they are doing and how they are doing with the body, they are communicating with their signals in the form of the metabolites. They are combining together with the microbiota, and they are making the microbiome, and they are communicating with the immune cells through their sensor, and they can change the outcome of the disease that may increase the disease, and that may decrease the disease. So in that form, then how these metabolites are sensed by the immune cells, because one of the most abundant metabolites in the body is the short chain fatty acids, and that is sensed by a range of the GPCR coupled receptors. They are present on the immune cells. So particularly But we need a model, experimental model. How we can create that model that more of the T cell will come into the picture and we can see the sensing between the metabolites and the T cells. Because in DSS model, that is more of the barrier function studies. But this is the hapten-based model that we channelize the cells, particularly TH2 and TH9, that will mimic the UC conditions with the humans. So in that particular case, we did the experiment and we just compare both the compartments of the UC patients in the experimental mice and with the human serum also. Among both the compartments, we found one of the key metabolites that was highly upregulated, and that was the succinate actually. We correlate with the sensing receptor that was present on the immune cells, pathogenic cells that was highly upregulated. It means both are in the picture, both are simultaneously present. So after that, we did the opposite of this experiment. We put the succinate from the outside. But when we put the orally, it is not accumulating in the gut colon because it is very small in size and got absorbed from the duodenum and all that part. But we need something that can locally increase the level of the succinate. So what we did, so in that case, we did the enema, more than 15 millimolar of the succinate, and we found a similar kind of pathology in those conditions. After that, we start thinking that how we can work on these nodes. Either we can work on the iron line, we work on the receptor, or maybe we can work on the succinate, and that can ameliorate the IBD condition. So after that, we just did the first experiment. We did the neutralization of the iron line, and we got that there was a decrease in the pathogenic cytokines levels or increase in the healing cytokines like IL-10. That is the anti-inflammatory cytokines. And there was amelioration in the IBD clearly. After that, we designed a blocker of the receptor that is with our collaborator, and we used the blocking of this receptor in the cells, and we got the same result. Expression of receptor is going down, and there was pathology was gone. So in that case, but why these particular cells are sensitized more? Because they have more expression of this particular receptor, the TH2 and TH9, because they are the major source of IL-9 in our body. After that, we start thinking that blocking of these two, we can ameliorate the IBD condition. But what about the succinate? So in succinate, we start thinking that either first we have to find out the source and other how we can block it. So after this, we did the experiment in the pseudo-germ-free mice, where we found that without the microbiota, there was no increase in the succinate level. After that, we did the FMT experiment. In those FMT experiment, in the disease one, there was a transfer of more succinate-producing bacteria in the gut, and there was elevation in the pathology as per that. So we can confirm. After that, we confirm the source. Actually, we found that vulgaris is the bacteria that is the key producer in the gut. And we also colonized this bacteria, and we checked the level of succinate that was high. After that, we correlate with the human settings also. We found that there was more Bacteroideaceae and less Acidomycococci. Acidomycococci is the consumer family and Bacteroideaceae is the producer family. So there was a disbalance of the ecosystem in those settings. After that, we start thinking that we got the source, this is the gut microbiota, but how we can block it? Then we start thinking that we can use something that can block the producer, or either we can increase the number of the converters. So we work on the vulgar test, and we found that there was a retinol sensor was present, and we put the retinoic acid. It will lead to the death of the vulgar test, and levels of the succinate will be gone like that. And we put the similar experiment, and on the basis of this information, we design a consortia that is producing retinoic acid in the gut. And we colonized this, and we did the similar experiment. We found that there was a decrease in the succinate along with the decreased amelioration of the condition. After that, we used the converter bacteria that can convert the succinate more into the short chain fatty acids. After that, we did the similar experiment, and we made a consortia using these two, and we colonized them. We tested in our setting, that is, they are working fine. They are decreasing the level of the succinate and all that. And overall, the summary is like, due to the dysbiosis in the IBD, there is a dysbiosis that is leading to the disbalance of the bacteria, and that is leading to the more production of the succinate, more of the proliferation of the pathogenic cells, and that leading to the more of the IBD condition. But by blocking these states, we can decrease the amelioration. Sorry, we can promote the amelioration of the condition. And finally, I would like to acknowledge our team at Teachers TI, my boss, and all our collaborators. Thank you. I'm ready for the questions.
Thank you very much, Radip. We can have one or two quick questions. It's very good information, the role of succinate, I believe.
Yeah, we got the patent.
So genera, species, and quantification also?
Yes,
How
yes.
much which one?
Yes, all together actually.
And this is all lab-based?
Yes, we isolated these strains from the human settings.
For GLP?
No, we are planning for that one actually.
You'll have to plan?
Yeah.
Okay, all right, good.
Any other?
Okay. Thank you.
Okay, thank you,
Thank you.
Radip.
I have a question.
What enzymes did you use to break the succinate to short chain fatty acids? What enzymes did you use to break succinate to fatty acids, more smaller fatty acids?
Not enzyme exactly. We use the consortia, bacteria.
Okay, the same
They are the
concept.
converter. Actually, in a healthy condition, it is like that there is a balance of the ecosystem. There are producers and consumers. And succinate is short-lived because not because of its chemistry, because of its demand actually, because it is the precursor for the making of the short-chain fatty acids. But if there will be no converter, then it will start accumulating. If once get accumulated, then through the SLC13 transporter, it will reach to the mucosa, and that will it will reach to the receptor carrying cells. They are pathogenic cells, and they will proliferate more. They will proliferate more cytokine. That will be the pro-inflammatory, and aggravating the disease will be like that.
Okay, thank you, Rajdeep.
Yeah.
Next.
Hello. I will now invite Mr. Akash Kumar Kharwar. He is from Naipur, Hyderabad, and is going to talk on L. plantarum and L. helveticus and propionic acid.
Good morning, everyone. So I would like to say thank you to the organizing committee to give me chance to present here. So the topic of my presentation is Lactobacillus plantarum and helveticus elevates the propionic acid and valproic acid generated autism spectrum rat model. So first, I would like to introduce about autism. So it is a neurological illness growing up day by day. It's increasing. It's around last past 30 years is 39%. So autism is characterized by stereotype behavior in which repetitive section we have seen, and less interested in the social communications. So in autism many uh, characters are there, like environmental factors, genetic factors, and metabolic factors. So we are focusing on this gastrointestinal dysfunctions, in which leaky gut syndrome we have found. So in current market scenario, we have seen that the probiotics is holds the promise, the potential therapeutics approach addressing the GI dysfunction in autism. So these are the prevalence rate of autism, in which you can see that the map in which darker one have more autism, and if you see the Indian map, you can see the prevalence rate of autism disorder. The scale is measured by the prevalence per lakh of population. So the rationale of this whole study is to check the different animal model help in studying autism like behavior, and this model allows the exploration of probiotic in ASD related phenotype and the mechanism. So in autism condition, dysbiosis happen. So this dysbiosis is further created like a neuronal alternation and this metabolite alteration, and with the help of with this probiotic, in which we use like plantarum and helveticus in our lab, we have done many of the studies like with different lactobacillus species like rotary, paracasi. But with the help of these two combination, we have found that this alteration happen in ASD condition with by this dysbiosis is balancing by with this combination of probiotics. So the main objective of this project is to investigate the impact of this both probiotic in propionic acid and valproic acid generated redfin. So this is the model we used for this autism. This first model is PPA model, that is propionic acid model. Second one is valproic acid model. So these are the result in which these are the behavior results. So first we have checked the repetitive and stereotype behavior with the help of open field test, and in this result you can see that the probiotic rescued this repetitive and stereotype behavior, and we have done this grooming test in which we have found that the reduce in the episodic of this grooming in treatment group. And this is the another behavior test which is MBT's marble burring test, in which we have found that the treatment of this both combination reduces the burring efficiency of this autism, right? And then we go for the anxiety related symptoms in autism. So with the help of this two combination of probiotic, we have found that the significantly reduce in the anxiety. uh, and this is the another behavior test in which we have checked the social interaction of this autism rat. So we have found that this probiotic have efficiency to improvement in the social interaction of this autism rat. And then we done the molecular part, like uh, pro information. We go for this pro information markers like IL-6 and IL-6 and TNF alpha. And with the help of this both probiotic combination, we have found that the increase in the pro-inflammatory marker in the autism rate is decreases by using of uh, this both probiotics combinations. And another, we have checked the oxidative stress in the plasma level in osteotic rat with the help of these two tests of SOD activity and catalysis test. Then we check for the metabolites with the help of the NMR in fecal sample. So we have found that the metabolite which are decreases with in autism through dysbiosis is restored with the help of this using this both probiotic. And after that, we have seen the morphologically in valproic acid model, this probiotic improve the intestinal morphology of this colon. So then we go for this tight junction protein, which are disturbed in autism through this dysbiosis. This both tight junction proteins are Z01 and occludin. So this probiotic all have effect on the restoration of this Z01 and occludin in autism rat. And we have checked this neurotransmitter like dopamine and GABA, which are decreases in.
Akash, come to conclusion.
So overall the conclusion of this study is VPA exhibit VPA rat exhibit autism like behavior, which is mimicking core feature of autism. And mm, this both lactobacillus plantarum and helvanthichus elevate the stereotype and anxiety behavior by enhancing the behavior function in gut microbial balance. Both probiotic reduce this pro-inflammatory marker and restore the neurotransmitter in PFC region. Okay, thank you.
Thank you. Any question?
Pardon.
Any strong scientific rationale for selecting these two species?
Ma'am,
Lactobacillus?
yeah, ma'am. So in our lab, we have checked for this rotary and this Paracasey and other lactobacillus also. But with the combination of this both plantarum and helvanthichus, we have found that the restoration of this pro-inflammatory and short-chain fatty is more as compared to both. We used single strains, like done single strain study like plantarum, singly plantarum and then helvanthichus. So these both show some concerta type of actions.
Okay, thank you very much, Akash. Now, next, I invite Anand EP from TCS Research Pune.
Hi, good afternoon. Thanking the organizers for providing me an opportunity to present my work here. So the work is titled Knowledge Graph Guided Personalized Probiotic and Dietary Recommendations for IBD. First of all, briefly introducing about IBD. IBD, as we know, is a multifactorial and complex disorder, which is influenced by the interaction between host genetics and gut microbiome. And this results in abnormal inflammation and, as depicted here, changes in gut barrier integrity, especially the disruption of gut barrier. And modulation of gut bacteria through factors like diet and probiotics. It can be a very effective solution when given in a personalized manner, and it could possibly help alleviating IBD symptoms. And several computational tools can be used to design these individualized approaches. Knowledge graphs is one such technique which can capture elegantly the heterogeneous interactions which happen in a complex disorder like IBD. So, going to the objectives of this work, firstly, the objective is to construct a gut bacteria-centric knowledge graph with a focus on gut bacteria to host associations. And secondly, developing a score that reflects the association of each bacteria with various host components that are reported to be involved in IBD. That is, factors which contribute to the development of IBD. And thirdly, to develop a workflow to design a personalized dietary and probiotic recommendation using microbiome data from individuals with IBD. So in methodology, we created a knowledge graph, which is basically a collection of all kind of association data in the form of triples or a subject, object, and a relationship between them. So the nodes involved in this knowledge graph include bacteria or gut bacteria at the center and host-related factors like host gene, diseases, food, and bacterial factors like other interacting bacteria, which could be probiotics, and bacterial functions, etc. So this knowledge graph was populated using data from curated public databases and also from patterns or data from experimental outputs from peer-reviewed journals. And biomedical text mining was also used for this purpose. And we have recently published a work in which we have given a methodology which can be used to take out interbacterial association data. Now, suppose we have a data from an individual, the clinical data as well as a microbiome profile. This data, we can see here, we have a list of bacteria which have been obtained from the person's gut. And the knowledge graph that we created called GBKG can be used to overlay on this list of bacteria so that we can create a matrix where we have a list of bacteria as the rows and we have all IBD associated host factors as the columns. So this could be IBD associated genes, immune factors, chemicals, and diseases. By diseases, I mean if there is any known association of the bacteria to ulcerative colitis, CD, or any other variations or different kinds of different stages of IBD. Now, this matrix can be populated again. So we see that this would be a very sparse matrix because most of the associations may not be available, or very few associations must be reported in the literature. And we generate another matrix using a method called collaborative filtering, where we try to fill in all values here. So basically, with this, these will be the predicted values which could denote the association of bacteria to those factors. And if we sum all these values across all the rows for each bacteria, we can generate a score which is called a gut favorability score. And if we scale it between 0 to 1, this would mean that a bacteria with a value closer to 0, GFS score closer to 0, would mean that it has low association to IBD associated factors, and a value closer to 1 would mean the association would be larger. Now, this association data and food association data from GBKG can be used to recommend food as well as probiotics. So in the first phase of recommendation, we can directly query the KG and use the GFS data to get a list of food and probiotic. And later, it can be modulated based on the person's preference, calorie, etc. So in results, we have constructed a very extensive KG, including a lot of diseases, and it's a broad KG. It is not IBD centric. And in this KG, we have nearly 25 million triples or edges and around 6 million nodes with 80 relationship categories. Now, this is an example graph which we obtained after querying based on representative bacteria. And this is how the relationships will appear in a graphical way. So we have a sample and we obtain the microbiome data, and after overlaying on GBKG, this is a representative graph. And in each of the bacteria in this will be given a gut favorability score, which can be used to give a recommendation. And for example, this is a sample recommendation which can be generated with this. So basically, in this work, we present GBKG, a knowledge graph, which is a very flexible tool, and it can incorporate gut bacteria association data and later can be developed to incorporate much more data. And using this, we develop a favorability score incorporating different host components that are associated to IBD. And clinical validation is, of course, necessary at each and every step of this. And this methodology can be actually incorporated into any mobile-based application, for example, to obtain more personalized clinical data of the person. And a combined recommendation of diet as well as probiotics, which could make use of this GBKG at some level, would actually be a good strategy that towards the treatment of IBD.
Thank you.
Thanks.
Any question?
I have one question.
Ah.
Actually, you have so many combinations, permutations, because different type of diets and foods and nutrients and different type of bacteria.
Yeah.
So how you will decide that which criteria will be in priority and which lower priority like that?
Yeah, so basically we can do a weightage like that, but in this case, we just provided a basic methodology. And we are not specifically giving any weightage to any of those
Is it
criteria.
targeted towards a specific application, maybe for IBD or maybe for immunity or like that?
So basically, in this context, we actually created a very broad KG, and parts of it, like you can target maybe a very specific disease, or you can target all diseases associated also. So it is up to the user. The user can query for a disease, like all the factors associated to the disease, and they can get it out of the KG and then use it according to whatever situation they are in. So that is in the importance of this kind of KG is that it is usable in multiple contexts, and this technology can be adopted to disease specific or broader way also.
Okay. Any other
Okay,
question?
I just want to ask a question here.
Yeah.
Yeah. So I think the concept is brilliant. You're trying to put all the variables together.
Yeah.
My query is from where have you taken the food database?
Okay.
And how are you arriving at the, recommendations for the diet? Because I saw in the last slide,
Yeah.
some recommendations were made.
Huh.
Can you please elaborate?
Yeah. So actually the availability of data is very low regarding exact bacteria to exact food correlation. So we had searched extensively about that, and we found a few literature evidences. So the sample data, what we showed here, was basically from that data. So it was just, I think, 2 sample papers which had kind of extensive data. And in that, the association to foods were at different levels also. So some of the foods were like very specific, like for example, a bacteria is known to be associated positively with red wine, for example. But there could also be associations like bacteria is known to be associated to group alcohol in a positive way or a negative way. So recommendation there is actually a bit tricky. So ideally it would require much more data, much more extensive data. So
So it
we need to
Cannot, it
collect
cannot be just food. It has to be the component of
component.
food.
Yes, yes. So we
Because
tried
when you
this.
talk about diet and
Yeah.
IBD,
Yeah.
it has to be more specific and precise.
Yeah.
So
So we did this manner also, like a bacteria is known to consume a carbon source or a nitrogen source or any such material. And this is a part of a diet or a food. And we had tried to build a relationship through that also. But the problem here is also that not even if a component is present in a food, it may not be necessary that the bacteria will utilize it at that moment in that patient, in that particular stage of the disease. So that is a very difficult problem to do. So obviously the recommendations, whatever we make through it, have a lot of context which is required. And one beauty of knowledge graph is that there are different graph databases in which we can load the data. So in some of the graph databases, for each relationship, suppose for example, Ruminococcus navis, we know it is related to MUC1 gene. So and that we know it is upregulated in IBD. And this is a relationship which we have, and this
Okay.
relationship can be represented.
Okay, Anand, thank you. I understood. Thank you very much. Now, next is Mithali Inamdar, National Center for Cell Sciences, Pune.
Hello. Yes. So very good afternoon, everyone. I'm Ms. Mithali Namdar. I am from National Center for Cell Sciences. I'm working with Dr. Dheeraj on mother-infant microbiome, and today I'll be presenting the same. Thank you, organizers, for giving me this opportunity. To start with, we all know that microbiome is an integral part of our body, and especially the infant microbiome is very, very important because it helps in development of various systems like gastrointestinal tract, the neurological as well as immune system. And ultimately, it helps in deciding the fate of our body. But any dysbiosis during this early acquisition of microbiome may lead to obesity, diabetes, colitis, or certain severe disorders or diseases. So this infant gut microbiome is affected by various, or there are many factors which are affected in the development of infant gut microbiome, is like the mode of delivery, the mode of feeding, etc. And there are various intrinsic factors also which help in development of this infant gut bacteria. For example, the HMOs. So human milk oligosaccharides are nothing but small sugar moieties which are present in the breast milk, which are not digestible by human body, but only digestible by infants. And which when digested by infants, these beneficial bacteria are proliferated, and that in turn leads to secretion or production of HCFAs like acetate, propionate, and butyrate. So I won't be showing this data today, but understanding the importance of mother-infant topic, we designed a project which initiated with the sampling of mother-infant cohort population. So we sampled 40 different mother-infant pairs, which resulted into 760 samples across 4 different time points. And we collected samples from mother's stool, skin, and breast milk. And from infant, we collected stool as well as oral swabs. So for all these 760 samples, we have done amplicon as well as shotgun metagenomics. This is the following workflow which we followed for amplicon sequencing, and I'll be showing you the results for amplicon sequencing now. So the initial results showed that the alpha diversity across different body sites in mother and infant were varying, and especially the mother's gut had the highest diversity. And we got the but obvious result that the infant gut microbiome is populating or is maturing over the time, which we can see in the alpha diversity. But the most interesting part which we saw was in the beta diversity. So here you can see the different colors are denoting the different body sites of infant and the mother. And you can see that the mother's gut and the infant's gut are having similar kind of diversity, which is shown in the beta diversity plot. So this really was an intriguing result in our analysis, because how do these gut bacteria are populated in the first time point itself in the infant? That means just after the birth. So you must have heard about yesterday's talk given by Professor Saurabh Datta that there are different ways in which infant gut is populated. So there are hypotheses like intraplacental, intrauterine, as well as the third and the most interesting one is the enteromammary pathway, which means in the enteromammary pathway, there are certain bacteria which can be seeped out from the gastrointestinal tract of the gut of the mother and get populated with the immune cells like dendritic cells in the breast milk, and which when fed internally or which when fed by infant are then populated by infant gut. So we started questioning, and then we further led to different kind of analysis to see whether this is really happening. So so we did a different kind of analysis called as cosine similarity indices. So in this, we have kept as infant gut one vector and another vector different body sites of infant and mother. And we saw that in the mother gut, so the infant gut was having a highest similarity with that of mother's gut across time point. Again, the same point was getting showed from the previous results itself. Then we further moved to source tracking. So we use source tracker 2 here to see what are the sources of infant gut bacteria, and is mother samples or are the mother microbiome contributing in it. So we can see here again that the mother gut has the influence, followed by the breast milk bacteria, which is there in the infant's gut at the time point 1, and eventually the breast milk samples contribution is getting lower and the mother's gut is getting increased, because you know that the infant's gut is also maturing. So taking hints from this, we further did the shotgun metagenomics, and there we found that the bifidobacterium strains are the most abundant strains which are seen in all the 3 samples. That means in infant gut, milk, as well as in mother gut. And then further, we took these mags and we did the strain tracing of these bifidobacterium strains, and we saw that the bifidobacterium bifidum, if you can see in the pink color, and the bifidobacterium longum are the 2 strains which are transmitted from mother gut to the milk to the infant gut. So now we know which bacteria is actually getting transmitted. But there are certain drawbacks to this study. And so these studies are basically the molecular based studies and usually done based on DNA technologies. So there can be a chance of contamination, or there can be certain hints that there can be a contamination. So we further decided, so I want to refer to yesterday's talk for this, that there are different kinds of pathways. So we further did isolation of bifidobacterium species from mother's gut sample, and we did transformation. So we transform M. cherry, that is a fluorescently tagged, red fluorescence protein, and we tagged it in the bifidobacterium, and we did animal studies. So in the animal studies, we gavaged this M. cherry tagged bifidobacterium from the pregnancy of the mice till the delivery of the pups, and we checked for the translocation of this bacteria. So there are different methods which we approached for checking this bacteria. So we did IVIS imaging. So IVIS is a whole body imaging. We did qPCR.
The conclusion.
Yes. So we did certain animal experiments. We found good results also. And the results are that from the infant's gut microbiome from birth is getting populated, and there can be also enteromammary pathway in the mother-infant dyads. But the last thing which I think was very important, the significance of the study, that I would again like to refer to yesterday's talk, that is Professor Patricia's talk, that she mentioned that there are various benefits which are there in the probiotic, but in the mother-infant population, which is very sensitive, there are certain drawbacks which are being seen in the recent studies. That is, the protocols are not standardized, the strains and the dosage is also not standardized. But I think if there is any mother-infant translocation, that this can be a very promising method through which the probiotics can be delivered, because the dosage of the strains cannot, we don't need to decide the dosage of the strain. Mother itself physiologically will decide the strain, dosage of the strain.
Thank you.
Thank you.
Any question?
So PYG plus blood plus rumen.
And you do the spread plating, right?
Yes, we did enrichment and spread plate.
And you kept in the anaerobic chamber.
Yes, we have used Don Whitley 835.
So if there is no question, thank you very much, Mitali.
Thank you.
Thank you.
The now last speaker is Sucheta Guleria from PGIMER, Chandigarh.
Good afternoon, everyone. The title of my study is Giardia infection is associated with altered community structure of murine gut microbiome post antibiotic and probiotic treatment. So starting with the introduction, giardiasis is a gastrointestinal parasitic disease which is caused by a protozoan Giardia duodenalis in humans. It has two forms, that is the trophozoite form and the cyst form, and the cyst is responsible for the transmission. And once the cyst undergoes excision due to the gastric secretions in the stomach, the trophozoites, they attach to the intestinal epithelial cells and they cause various gastrointestinal symptoms. So giardiasis is associated with acute and chronic diarrhea in children and adults worldwide, and also result in post-infectious complications, that is irritable bowel syndrome and chronic fatigue syndrome. And interestingly, 75% of the infection is asymptomatic. So the exact mechanism behind giardia pathogenesis remain incompletely understood, but recent research underscores the crucial role of the microbiota. As we already know that the intestinal microbiota, it plays a critical role in the gut homeostasis and overall health, and its alteration by the entropathogen is associated with the gastrointestinal diseases. And also the dietary interventions, they contribute to the microbial diversity and it influence the giardia pathogenesis. Therefore, understanding the role of specific commensal bacteria during the infection could help in exploring new treatment options and monitoring therapies such as fecal microbial transplant. So the objective of my study is to evaluate the shift in the community composition of the gut microbiome associated with giardiasis in untreated and pre-treated, that is pre-treated with antibiotics and probiotics in mouse model. So coming to the material and methods, so the study includes 168 biopsy mice. They were further divided into 3 groups. The first group was treated with antibiotics for 4 days by oral gavage. The second group was treated with the lactobacillus rhamnosus, that is the probiotic, for 7 days by oral gavage. And the third group was not treated with antibiotic or lactobacillus. Now each group was further divided into 2 subgroups. The one subgroup was infected with the giardia trophozoites, and the other subgroup acted as its respective uninfected controls. And mice in the replicates of 8, they were sacrificed at 3 time points, that is the 3, 7th, and 14th day post-infection. So for the giardia infection, we have maintained the azeni culture of this parasite in our department. In the TYS33 media, you can see the culture tubes. These are the tubes in which the parasite is maintained. And the figure also shows the trophozoites under the 10x magnification and the trophozoites in the iodine as well as the wet mount under 40x magnification. This is how we have given the animal inoculation of the giardia trophozoites as well as the probiotics. And the lactobacillus rhamnosus has been maintained in the MRS agar plates. So for the assessment of the microbial diversity, the luminal contents were extracted. They were isolated, DNA was extracted, and V3, V4 region of 16S was amplified. And also to actually study the impact of these pre-treatments on the giardia trophozoites, we did the quantification using the quantitative PCR. So coming to the results, as we can see from the first diagram, the swelling of the large intestine actually shows that the antibiotic lead to the dysbiosis of the commensals. And to confirm the establishment of the lactobacillus infection, we performed the lactobacillus specific PCR. And to confirm the establishment of the giardia infection in the mice model, we actually performed the nested PCR targeting the TPI gene of giardia. So coming to the microbiome analysis, we can see that the antibiotic pre-treated and untreated group, they lead to the increase in the phylum Proteobacteria, Campylobacterota, and decrease in phylum Firmicutes up to 14-day post-infection, in contrast to their respective uninfective controls. Whereas in probiotic pre-treated group, it leads to decrease in the phylum Proteobacteria and increase in the phylum Firmicutes up to 7 days, and decrease in phylum Campylobacterota up to 14-day post-infection, similar to respective uninfective controls. So as you can see from the alpha diversity, the antibiotic pre-treated group, it basically remained consistently lower at all the time points, indicating that the dysbiosis of the antibiotics along with the GRD acid, it further disrupted the commensals as compared to the untreated ones, whereas the probiotic treated group, you can see the alpha diversity is higher at all the time points, indicating that the integrity of the composition of the gut microbiome, it was intact regardless of the Giardia infection. From the qPCR analysis also, we can see that the Giardia infection actually led to the infection intensity, increase in the infection intensity at 3rd day, peaking at 7th day and further decline at 14th day. But when we compare to the antibiotic pre-treated group, we can see the 10-fold increase in the Giardia quantification. And on the other hand, treating it with probiotic, that is Lactobacillus rhamnosus, we can see the gradual decrease from 3rd to 14th day. So coming to the discussion, as I've already told that the Giardia duodenalis, it leads to the increase in phylum Proteobacteria, Campylobacteriota, and decrease in firmicutes up to 14th day, whereas Lactobacillus pre-treated group shows the opposite trend with increase in Proteobacteria and firmicutes, but only up to 7th day post-infection. I wanted to highlight a point here that it leads to the contrasting results only up to 7th day, suggesting that the regular dose of probiotics are required for maintaining the gut flora. Whereas the Shannon and Simpson diversity indices, they were also consistently lower in the antibiotic as well as untreated mice. Whereas the probiotic fed group, they exhibited the highest diversity. And it basically suggests that regardless of the Giardia infection, the microbiome remained intact at all the time points. And moreover, from the RT-PCR analysis, the antibiotic pre-treatment exhibited a robust infection rate, and the lactobacillus administration, they reduced the parasite burden at all the time points. So basically, the study found the potential danger of the antibiotic usage causing the greater disruption of the gut microbiota as depicted by the alpha diversity, and resulted in more severe and prolonged Giardia infection as compared to untreated group, whereas the mice pre-treated with probiotics, the microbial diversity remained intact regardless of Giardia infection. Also, it led to the reduction of the parasite load, mitigating both the severity and the duration of the infection. So key message, the study emphasizes the importance of the intestinal microbiome against Giardia infection. These findings suggest that antibiotics usage during this infection could lead to negative health outcomes, and the study provide potential avenues for the probiotic therapy and help in monitoring strategies such as fecal microbial transplants. Thank you.
Thank you. Any question?
Yeah.
Yes, please.
By not using the right antimicrobial. Your study said that in the first group you use ampicillin, neomycin, and I don't know what all.
Vancomycin,
Vancomycin.
Yes.
For Giardia, treatment is using metronidazole, tinidazole, ornidazole, or you have the new one, which is nitazoxanide. So why did you use this group? Because you're starting with treatment with antibiotics for a disease which is not going to work.
Actually,
And then you compare it by giving probiotics and say that this is the best way, the results are good. So that's why this
sir,
is the point.
actually, the metronidazole and tinidazole are the drugs for against giardiasis, but as it causes diarrhea, that is acute to chronic diarrhea, it is misdiagnosed and people take antibiotics for diarrhea. So to cause the dysbiosis in the mice model, these particular antibiotics, they were used in order to
I'm saying if you had used metronidazole, it would have given the result which would have been comparative, that whether the probiotic is as effective as metronidazole or not.
yes,
Here
so
you
we can
compared it
add
only
the
with
one group
antibiotics.
also of the drug also.
Okay. If there is no other question, I think I'll invite jury to declare the results.
Thank you. Thank you.
Thank you so much.